Bacterial genomes consist of a single haploid chromosome and extrachromosomal elements such as plasmids and bacteriophages. Remember, haploid means that it only has half the total genetic code, or N. It also follows that diploid means that the organism has the full complement, or 2N. These genetic elements may be independent of the bacterial chromosome and can be transferred from one bacterium to another via transformation, conjugation, or transduction. Transformation occurs when bacteria take up free DNA from the environment and begin to express it. Those of you who have done some molecular biology research in the lab may have done bacterial transformation before. You take your plasmid that you want to clone, and you mix it with the bacteria. Here's your E. coli. You mix these together in a tube, and you temporarily disrupt the membrane using either heat shock or electroporation, and this allows the DNA to be taken up by the cell. Basically, something to allow the DNA to penetrate this normally intact bilayer. Then the bacteria are recovered and allowed to grow. Conjugation occurs when genetic material is transferred from a donor bacterium, in this case the F+ male, which carries the plasmid, to the recipient bacterium, the F- female, which does not contain the F plasmid. The F plasmid itself carries all the genes necessary to promote its own transfer. E. coli is an example of a bacteria that undergoes conjugation. Now, when the F plasmid becomes incorporated into the bacterial chromosome, the bacteria becomes a high-frequency recombination cell, or an HFR cell. The HFR cell now has the ability to transfer whole pieces of chromosomal material into a recipient bacteria. Transposition occurs when bits of DNA are excised and then reincorporated at another location. So let's say we have our gene of interest here. We're going to leave it blank, and we have a small flanking piece of DNA on either side. And let's just say, for the sake of argument, that it's going to get incorporated directly in the middle of this gene right here. So what's going to happen is you're going to have your original piece of DNA. Now we're going to have that gene, and then next to it is going to come the gene that we excised, and then the flanking segment, and then the gene that was cleaved, and the rest of the DNA. Great. Now, this can be incorporated to or from plasmids or chromosomes. Sometimes the excision is not precise, and flanking DNA can be incorporated, as I illustrated. Plasmid DNA can be transferred to another bacterium, and this is actually how much of the drug resistance in bacteria occurs today. Transduction can be generalized or specialized. This image shows the mechanism of transduction. In general, genetic material is transferred by bacterial viruses known as bacteriophages, right here. A lytic phage will infect the bacterium, leading to cleavage of the bacterial DNA and synthesis of viral proteins and viral DNA. Essentially, the bacterial cell gets hijacked by the bacteriophage, which selfishly uses the bacteria to make its own DNA. Parts of bacterial chromosomal DNA may become packaged into viral capsids instead of the viral DNA, and the phage can then transfer those genes when infecting another bacterium. Specialized transduction is an excision event where a lysogenic phage infects the bacterium, but the viral DNA actually incorporates into the bacterial DNA, as you can see here. When the phage DNA is excised for packaging, flanking bacterial genes may be taken with it. There are 5 toxins which we've already discussed that are encoded in a lysogenic phage, and to remember them, I like the ABCDE mnemonic: Shiga-like toxin, botulinum toxin, cholera toxin, diphtheria toxin, and the erythrogenic toxin of Strep pyogenes. In these examples, the bacteria are non-toxic until they become infected with a lysogenic phage carrying the gene for that toxin. For example, Corynebacterium diphtheriae only produces diphtheria toxin after it's been infected with the phage carrying the gene encoding the diphtheria toxin. This brings us to the end of the basic bacteriology lecture.